have. Let us see the content to be written in the biology practicals during the practical session. First major experiment A. This is like 1 mark. Requirements 1 mark. Procedure 1 mark. Okay. Observation 1 mark. Precaution 1 mark. Total 5 marks. This is the contents. Okay, take a screenshot. Next is mind experiment. To study the plant population density by quadrat method or frequency by quadrat method, anyone can be there. Okay, so for this principle compulsory 1 mark, requirements 2nd mark, procedure 3rd mark, observation conclusion 4 marks. Okay, so this is very important. This is the notes. Okay, next either this that one. To study the plant population frequency by quadrat method. Same principle, 1 mark, requirements 1, procedure 3rd mark, observation, okay, 4th mark, combined with conclusion. Okay, the third experiment notes. Next slide preparation. Okay, first experiment, pollen tube growth and stigma. Okay, aim, principle, requirements, procedure, observation, precaution. It's very short. Fifth experiment, mitosis in onion root tips. Principle, requirements, procedure. This is a procedure you have to follow. Okay. Next, preparation slide. In most of the cases, they'll ask this. This you have to write separately. That's why this will be given 5 marks. Study of slide. What you observe, under which magnification you observe. Observation. Okay. You have to write specially observation. That is, any phase, prophase, metaphase, anaphase, telophase. But metaphase writing is much better because this only can be easily identification possible. Okay, only this can be identification can be done. So metaphase is much better, remember. But they may ask any phase to write also. So this is very careful. Okay. So remember, anything you can write, but metaphase is much better. Okay, preparation of the slide. Procedure. Here, recheck. Procedure. Preparation of the slide. And the slide details what you observe. This is about the major, minor slide experiments. Now next is spottings. 7 spottings they'll give in the, you have to write like this. Even spot is identified as. Reason for identification of spot is observed characters, features. Okay. Now for your idea, I have made notes. See, this is the details of the every spot. Okay. I think all spottings have been covered. Take screenshots or just write it down. Flowers pollinated by wind. This is the spot. This is the image they will give. Okay. Reason, less weight and dry pollen. Here you observe that pollen grains are moving the air in the wind. This is the reason. And characters. These characters you can remember from the theory also. Not only from here, anyway. Next is spot is this one. Insects as pollinators. Flowers pollinated by insects. Why? Because pollen grains are attached to its body. So pollen is sticky, large, and heavy and rough. So like this reason you have to mention. Generally, most of people may not ask, but some cases, some externals will ask. Then you have to write. Why? Because 1/4. Okay, here 1/2 mark. So like this, 1 mark will be divided into 4 parts. Half plus half. Third, okay, flowers pollinated by birds. Reason may be because of the bright colored flowers. Okay, and characters. So remember this. Next is germination and stigma through permanent slide. A small pollen tube is seen through the style. Okay. Next is characters. These are the characters we have to remember. Next, this is for gamete development in the testis. Large number of seminiferous tubules. Reason, characters. This is ovary, gametes, development in ovary. Cell with follicular layers, primary, secondary, tertiary. Characters. Note it down. Meiosis in onion bud cell of grasshopper testis. Because a stage of meiosis they have observed. So this is the characters. TS blastula through Turner slides of mammals. Here blastula with a cavity. The cavity. Characters. Mendelian inheritance. This image they'll keep different seeds with the round and wrinkled shapes. Okay. Color also they can keep, but that's very rare. So these are the characters. So remember, these are the characters. Next is the pedigree chart. Pedigree chart will be kept. So rolling tongue, blood groups, earlobes, widow's peak, color blindness, anything they can ask. Okay. So these are the traits, autosomal traits. In this, 1, 2, 3, 4. These 4 are belongs to autodominant trait. Remember this. Okay. Color blindness, sex linked recessive. Okay, this is auto, sex. Is dominant, is recessive. Okay. X linked recessive trait alone, color blindness. This is an easy question they may ask. Next is emasculation, because anther is dissecting. Characters. Tagging. Label on the female flower. After emasculation, that will be considered female flower. So it should be mentioned like emasculated, particular date, plant, experiment number. So that is the tagging. Bagging. Okay, buttercup paper will be a plastic bag will be covered. This is the bagging. Reason. Understood? Characters. Ascaris. External features of roundworm, because it's looking like a roundworm. Then characters. Entomoeba. Pseudopodia. It belongs to amoeba, right? Characters. Because pseudopodia was observed like this. Plasmodium vivax. A vacuole with the nucleus at one side of the cell. Special. So it looks like a ringworm. Wrong. One side nucleus. Characters. Fungus. Hyphae cotton-like ringworm. Characters. Root nodules. Round bodies in the roots. Characters. Cuscuta on host. A close association of the climber to the stem, on the stem. Characters. Lichens. White filaments and greenish thallus. Okay. Characters. Homologous organs. Okay. Bones are similar in the structure. Characters. So this is a homologous organ, image they may keep, or analogous organ image they may keep. Analogous organs. Bones are different in structures but similar function. The characters. Okay. Also, we should have an idea about the microscope and its parts. It will be helpful. Okay. Remember different parts you should remember. Hope you understood. Thank you. Have a nice day.